Okay, let's dive right into this explainer on the living world. Have you ever looked around at the sheer, overwhelming variety of life on Earth? I mean, from the microscopic bacteria sitting on your skin right now to the absolutely massive trees in a rainforest. Have you ever wondered how on Earth scientists actually make sense of it all? Well, today, we're going to look at how scientists, both early and modern, took the absolute chaos of nature and somehow transformed it into a perfectly ordered, universal system.
Here's our roadmap for this explainer. We'll quickly hit the scale of biodiversity, move to the universal naming system, explore the hierarchy of life, check out some real-world classification examples, and then we're going to test your knowledge with some previous year questions.
Section 1. The Scale of Biodiversity. Grasping the Biological Chaos.
1.7 to 1.8 million. Just think about that number for a second. That is the current estimate of the species we've known and described so far. It's our baseline for biodiversity. And honestly, this is just what we've found up to this point. As we explore new environments, or honestly even double-check the old ones, new organisms are constantly being identified. This massive number, that is the sprawling biological chaos we're trying to organize.
Now, a massive shout-out has to go to Ernst Mayr. He was an evolutionary biologist from Harvard, and he's often referred to as the Darwin of the 20th century. Over a crazy long career spanning nearly 80 years, he almost single-handedly made the origin of species diversity the central question of evolutionary biology. He also pioneered the current definition of a biological species, giving scientists the fundamental building block they really needed to start sorting through those 1.8 million organisms.
But hey, before we can classify life, we actually have to define it, right? And this is exactly where entrance exams love to try and trick you, because not all traits displayed by living things are considered defining features without exception. Take growth, for example. Is growth a defining feature of life? Actually, no. Why? Because of extrinsic growth. Think about it. Non-living things like mountains, boulders, or sand dunes, they technically grow by accumulating material on their surface. Living growth, though, is intrinsic. It happens from the inside. Plus, if you look at unicellular organisms like an amoeba, growth and reproduction are practically the exact same thing. They both just mean an increase in the number of cells.
So the crucial point here is that we absolutely must separate the defining features from the non-defining ones. Reproduction, not defining. A lot of living things just can't reproduce. Think about mules, sterile worker bees, or infertile human couples. Metabolism, on the other hand, is a defining feature. There's literally no non-living thing that exhibits metabolism. Cellular structure, absolutely defining. No non-living entity has cellular organization. And finally, consciousness. This is the most technically complex defining property of all living organisms. Though, you gotta remember, humans are the only organisms with self-consciousness, which creates some pretty unique medical edge cases, like patients in a coma on life support.
Section 2. The Universal Naming System. Cracking the Scientific Code.
So now that we know what life is, how do we talk about it without getting completely lost in translation with local languages? We need a universal system. Enter binomial nomenclature. This was a brilliant system developed by Carolus Linnaeus, and the international rules for it are super strict. First, every name has two parts, the generic name and the specific epithet. Second, the language has to be strictly Latinized no matter where the organism actually comes from. Third is formatting. Names are always italicized in print, but if you're running them out by hand, you have to underline both words separately. And finally, the rule of priority. Basically, the earliest validly published name after Linnaeus' 1753 publication is the official one. It's the ultimate rule to prevent global confusion.
Now, what's really interesting on this slide is a quirky little exception called tautonyms. This happens when the genus and the species names are exactly the same. For instance, the scientific name for the Indian cobra is Naja naja, or the rat, which is Rattus rattus. But here's the catch you definitely need to remember. Tautonyms are perfectly fine in zoology for animals, but they're strictly banned under the rules of botany for plants. Exam writers love to test that little detail, for sure.
Section 3. The hierarchy of life, sorting into the biological filing cabinet.
Let's move to the taxonomic hierarchy and see how this actually builds, step by step. Think of it like a descending filing cabinet. It starts way at the top with kingdom. That's the highest category, it's super broad, and its members share the least amount of characteristics. Then we step down into phylum for animals or division for plants, then down to class, order, family, genus, and finally, species. Now, as you go lower down these taxonomic ranks, the number of shared common characteristics among the organisms drastically increases. A species is the lowest taxon. It's a very specific, concrete biological unit.
To help scientists figure out exactly where an organism goes in this massive filing cabinet, they use taxonomical aids. A herbarium, for example, is a storehouse of dried, pressed plant specimens preserved on standard sheets. Botanical gardens are specialized collections of living plants for reference. Then you have a taxonomic key. This is an identification tool based on a list of contrasting characters, usually in pairs that we call couplets. Each individual statement in that key is called a lead. And finally, monographs. These are books that contain highly detailed information on just one specific taxon.
Section 4. Real-world classification examples. Let's take a mental screenshot here because this is our NCERT Table 1.1 fast recall.
All right, looking at this master chart, let's make the theory concrete. Look closely at the housefly. Biologically, it's Musca domestica. The genus is Musca, the family is Muscida, order Diptera, class Insecta, and phylum Arthropoda. Now compare that to a plant, like wheat or Triticum aestivum. Genus Triticum, family Possaceae, order Poales, class Monocotyledone, and division Angiospermae. Notice the suffixes here. Families in plants often end in ACEAE, while orders end in ales. Memorizing these specific pathways is going to be incredibly useful for recognizing patterns later on.
Section 5. Test your knowledge. PYQs. This is the final boss fight. Let's see if you can apply what we just talked about. Challenge number 1. Which two of the given points are known as the twin characteristics of growth? Is it A, increase in mass, B, increase in number of individuals, C, cellular organization, or D, response to stimuli? Think about it for a second. If you guessed A and B, you nailed it. An increase in mass and an increase in number of individuals are the twin characteristics of growth. Remember that unicellular amoeba example we talked about earlier? Increasing in number is essentially the exact same as growing for them. Okay, moving on to challenge 2. Nomenclature is governed by universal rules. Which of the following is contrary, meaning completely against the rules of nomenclature? Option A. Biological names can be written in any language. And this brilliantly illustrates why knowing the specifics is absolutely crucial. The correct answer is A. It is totally contrary to the rules to write biological names in just any language. They have to be strictly Latinized or derived from Latin, regardless of where they come from. That's the whole point of a globally functional system. All right, final challenge. Let's look at a highly tested edge case. True or false? Statement. Isolated metabolic reactions in vitro, meaning inside a test tube, are living things. What do you guys think? The answer is absolutely false. It's such a vital nuance. The source material explicitly says that isolated metabolic reactions performed in vitro are just isolated chemical reactions. They are not living things. They are definitely living reactions, but without a cellular structure, a reaction in a test tube just can't be classified as a living thing. Which brings us to the end of our explainer. We've done a lot of categorizing and filing today, but I want to leave you with this thought. If the 1.8 million species we've painstakingly classified is just a fraction of what is actually out there, what kind of bizarre, completely unimaginable exceptions to the rules of life are currently waiting to be discovered right now in the dense jungles or deep oceans? The biological filing cabinet is nowhere near full, guys. Keep learning, keep exploring, and I'll catch you in the next one.